Hello everyone. In last two videos of this series, I have already discussed in detail about the introduction of genetics and biography of Gregor Johann Mendel. Now in this video, I am going to focus on different terminologies that are mainly used in genetics. Okay, so total 12 different things that we are going to discuss in this video. So first is character. Okay, so what is character? Character is nothing but the feature of living organism. Okay, so it is the feature of organism. For example, if we consider Pisum sativum or garden pea, which is selected by Mendel for his experiment, there are 7 different characters which Mendel focused. Okay, like height of the stem, that is height of the plant, then size and shape of the seed, size and shape of pod, flower color and flower position. So all these are nothing but the characters. So it is nothing but the feature of organisms. Okay, now trait is similar to the character. Only difference in between character and trait is that traits are the inherited characters. Okay, so they are nothing but the characters, but which are inherited from one generation to another generation. Okay, for example, if we cross one tall pea plant with another dwarf plant, in that case in F1 generation or in first generation, all tall plant will be there. Okay, so this tall character is transferred from parent to offspring, and now it is called as trait. Okay, so traits are nothing but the inherited characters, and example remains same, like tall plant produced in F1 generation, okay, or first generation, which represent the tall dominant parent plant. Okay, if out of tall and dwarf parent, tall is the dominant parent, in that case in F1 generation all the plant will be tall plants. Okay, and now this tall is nothing but the trait because it is inherited character. Okay, now the third thing that we frequently noticed in Mendel's experiments is factor. Okay, so this factor term is coined by Mendel himself, and the factors are nothing but the particles which are present in the organism, and these particles are responsible for inheritance and expression of characters. Okay, so any character like height of the plant or color of the seed or shape of the seed transfer from one generation to the next generation through factors. So factors are nothing but the particles which is present in the organism, and these factors are responsible for inheritance, that is transmission of the character, and expression of the character in progeny. Okay, so inheritance and expression of the character is actually performed by factors. Okay, characters are not as it is transferred from generation to generation, but factors are responsible for transferring character from parent to progeny. Okay, so this term is coined by Mendel. Similarly, the gene term, and this gene term is coined by Johansson, and gene and factors are similar. We know that Mendel's factors is nothing but our genes. Okay, so genes are the segment of DNA. Okay, and they are also responsible for inheritance and expression of characters. So genes are responsible for inheritance and expression of characters. Okay, for example, if I draw double stranded DNA structure here, okay, so this is the strand 1 which running from 5 prime to 3 prime direction, and the anti-parallel strand which run from 3 prime to 5 prime direction. Okay, this is the Watson and Crick model of the DNA. Now the DNA having nucleotide base pairs. Okay, suppose we select this portion of the DNA, and if this portion of the DNA is producing a functional protein after transcription and translation, and this portion which produces the protein is responsible for expression of certain kind of character in the organism, in that case this particular segment of the DNA is nothing but the gene. Okay, so gene is the segment of the DNA, and this gene term is coined by Johnson. Okay, and this fragment of the DNA is also responsible for inheritance and expression of character in the organisms. Okay, so we have discussed 4 terms now, that is character, trait, factor and gene. Characters are nothing but the feature of organism, then traits are inherited character, factors are the particle which is responsible for inheritance and expression of the character, while genes are nothing but the factors, but they are the segments of the DNA. Okay, so now the next or character number 5 is alleles. This term was coined by Bateson, and alleles are nothing but the one or more alternate forms of gene. Okay, so if the homologous chromosome have 2 different type of genes at a same location and both coding for a similar character, in that case these 2 genes are nothing but the alleles. So according to Bateson's, one or more alternate forms of the gene present at identical location on homologous chromosomes are alleles. Okay, so initially scientists were believed that actually there are 2 different type of alleles present in the organism, but nowadays we know that many characters are encoded by allele more than 2. Okay, it is called as polygenic inheritance. Okay, so if we draw 2 homologous chromosomes here, what is homologous chromosomes? The chromosomes which are morphologically and functionally similar. Okay, which is present in diploid cells. Now suppose these 2 are the homologous chromosomes. Now the first chromosome have 1 gene at this position, and the second chromosome suppose have similar kind of gene at this position. Now both these genes encode same character, for example height of the organism. In that case these 2 genes are called as alleles. Okay, so now you can connect the diagram with the definition of the allele. Alleles are the alternate forms of the gene which is present on homologous chromosome at identical position. Okay, in many organisms nowadays scientists have reported allele more than 2. Okay, so this is about alleles. Now what is dominant allele and what is recessive allele? So these 2 terms are used for the alleles now. Actually dominant allele is a type of allele which can express in the organism in both homo as well as heterozygous condition. Okay, what is homo and heterozygous condition? For example, a garden pea plant have capital T alleles. Okay, so both this type of genes is responsible for height of a stem in the garden pea plant, and if the pea plant have 2 capital gene, in that case height will be tall. The plant will going to have tall stem. Now if another plant have 1 capital T and another small t genes, we know that this small t is for dwarf phenotype. and this capital T for tall phenotype. Now in this case, the condition become heterozygous. Okay, if the organism have both the allele which is similar like this, it is called as homozygous condition. And if the organism have two alleles which are dissimilar, then in that case it is called as heterozygous condition. And now in heterozygous condition, one dominant allele is present, that is capital T, and another recessive allele is present which is called as small t. In that case, this capital T or dominant allele will going to suppress the effect of dwarf or recessive allele. In that case, the plant height will going to become tall. Okay, so height remain same, tall, but the genotype changes. One is homozygous, another is heterozygous. Okay, so this capital T is nothing but the dominant allele which can express in both the condition. It can express in capital T, capital D, that means homozygous condition, as well as this allele, dominant allele, can express in heterozygous condition, that is capital T, small t. On the other hand, next thing is recessive allele. Okay, this allele can only be expressed in homozygous condition. Okay, so recessive allele cannot express in heterozygous condition, okay, like dominant allele does. Okay, so now if we take the example, this small t is for dwarf phenotype. So if the plant have small t genotype, that means two alleles and both are same, that is small t, that means homozygous condition. In that case, the height will be dwarf. Okay, plant will have dwarf height. But if this T is present along with the capital T like this, the height will become tall. Okay, because this small t is a recessive allele which cannot express in heterozygous condition. When this small t is present along with the dominant allele, that is capital T, the effect of small t will going to repress. Okay, in that case only dominant allele will express itself. But when this recessive allele are present in homozygous condition, if the organism have both recessive allele, in that case they will express their phenotype like dwarf. Okay, if the seed shape in garden tree plant is round and wrinkle, which is denoted by word capital R, okay, or letter capital R, then if the organism have capital R, small r, in that case seed shape will be round. But if the organism have small r, now this small r is recessive and it can express itself and the shape become wrinkled. Okay, so remember that dominant allele can express itself in homo as well as heterozygous condition, because this dominant allele will going to suppress the effect of recessive allele. But a recessive allele can only be expressed itself in homozygous condition. Okay, that means both the allele should be same, and then only this type of allele can express the phenotype. Okay, now dominant alleles are always denoted by capital letter. Okay, they are always denoted by capital letter, like capital T for tall, capital Y for yellow, capital R for round shade. Okay, so all these type of dominant alleles are denoted by capital letter, while recessive alleles are denoted by small letters, like small t for dwarf, okay, then small r for wrinkle shape of the seed, and small y for green color of seed. Okay, so recessive alleles are denoted by small letters. Now the next thing is phenotype. What is phenotype? Phenotype is nothing but the external appearance of the organism. Okay, how exactly organism look from outside? Okay, it is nothing but the phenotype of the organism. For example, if we consider height of the pea plant, in that case there will be two different phenotypes like tall and dwarf. Okay, one plant will be tall while another is dwarf, and both these are the type of height of the plant. Okay, type of the plant on the basis of height. So we can call both this type or both this character as a phenotype of the organism, because we can easily observe this thing externally. So phenotype is the external appearance of the organism. On the other hand, genotype is the genetic makeup of the organisms. Okay, if we study the external characters of the organism, it is the phenotypic study, and if we study the internal organization or genetic makeup of the organism, we can call it as a genotypic study. Okay, so the term 5 to 8 is alleles, which is coined by Bateson, which is nothing but the one or more alternative forms of the gene present on identical loci on homologous chromosome. Then dominant means the type of allele which can express in homo as well as heterozygous condition. Recessive means type of allele which can only express in homozygous condition. Then phenotype means external characters or external appearance of the organism, like in case of height, two different phenotypes, tall and dwarf. Okay, now the genetic term number 9 that we will going to discuss in this lecture is genotype. So genotype is the study of genetic makeup of organism. Okay, so if we consider now tall plant, in that case the genotype of the tall plant in garden P will be capital T or capital T small t. Both these type of genotype will going to give tall plant. So the genotype of the tall will be capital T or capital T small t. So these two are the genotype for tall. Similarly, the genotype for the dwarf plant will always be small t, small t. Okay, when we consider genetic makeup of the organism, it is called as genotype. Okay, so two different genotype for dominant character, that is tall, while single genotype, that is small t for dwarf. Same thing will be there for all other 6 characters of the garden P, which Mendel select for his hybridization experiment. Okay, so now the term number 10 is homo and heterozygous organism. Okay, what is homozygous and what is heterozygous? It is clear from this thing now. If the organism have two allele which is similar, it is called as homozygous condition. Okay, if the organism have two similar allele, it is called as homozygous condition, like capital T or small t. This capital T or small t. It is called as homozygous condition. And if the organism have two dissimilar allele, then it is called as heterozygous condition, like capital T, small t. This. Okay. Now, homozygous organism like capital T or small t always produce single type of gametes. Okay. For example, now this capital T type of organism will going to produce single type of gamete, that is capital T, while small t type of organism will going to produce gamete small t. Okay. On the other hand, heterozygous type of organism produce two different types of gamete, like this organism will going to produce capital T as well as small t, two gametes. Okay. We know that the second law of Mendel, law of independent assortment or law of segregation, tells that the alleles get separated at the time of gamete formation. So this capital T will going to produce one gamete, while the small t also going to produce another gamete. So heterozygous organisms produce two different type of gametes, while homozygous organism always produce single type of gamete. Okay. Now the second last term of today's video is F1 generation. Okay, this F is nothing but the filial one generation. The filial means offspring produced from sexual reproduction. Okay, so any progeny which is produced after sexual reproduction is nothing but the filial generation. Okay, if we cross one tall plant with another dwarf plant, pure tall plant with dwarf plant, in that case F1 generation will going to show all tall plant. Okay, and it is called as F1 because it is the first generation. It is the Latin word filial, which means son. Filial means son. Okay, and also known as offspring produced by sexual reproduction. If we cross the plant which is produced in F1 generation, in that case they will going to give F2 generation. Similarly, there is F3, F4 generation. Okay, and the number continues. So F means filials, which is nothing but the offsprings that are produced after sexual reproduction. Now the next and last term which is used in the genetics frequently is Punnet square. What is Punnet square? It is the type of a diagram which is discovered by R.C. Punnet. Okay, now this scientist developed this Punnet square, also called as checkerboard, and this Punnet square type of a diagram is used to determine all the possible genotype and phenotype which is present in any cross. Okay, for example, if we cross one heterozygous plant capital T with another plant capital T. Okay, so we cross one F1 generation plant with the another F1 generation plant. It is called as selfing. Now in this case we can determine all possible genotype and phenotype of this cross by using Punnet square. Okay, now we have to just draw a simple diagram here. Now the first organism, suppose this is male and this is female. Now the first organism will going to give two type of gamete, capital T and small t. Similarly, the female will going to give same gamete, capital T and small t. So we have to just write the gamete at this position in the Punnet square, and now we can get all the possible genotype and phenotype, like at this point capital T, capital T small t, capital T small t and small t. So we can easily determine the genotype here, capital T 1, then capital T small t 2, and last small t. So we will going to get three different type of genotypes in the ratio 1:2:1. And from this genotype we can identify phenotype of the organism as this capital T will be tall, then this 2 capital T small t will be tall because of the dominant, and the last small t will be dwarf. So with the help of Punnet square we can easily determine genotype 1:2:1 and phenotype tall and dwarf. Okay, so Punnet square is used by breeders to identify all the possible genotype and phenotype during the crosses. So last we have discussed genotype, that is genetic makeup of the organism, homo and heterozygous type of organism, F1 generation or filial 1 generation, and Punnet square, which is developed by R.C. Punnet to determine the all possible genotype and phenotype. So total 12 different genetic terms we have discussed in this video. Now in the next video I am going to talk about 7 different characters which is selected by John Mendel in pea plant. Thank you.